gives me great pleasure to introduce Dr. Frevert from Berlin, Germany, who will share with us the various types of botulinum toxins and how they are similar and how they differ. Let's welcome Dr. Frevert in the usual way. Thank you.
Thanks for the introduction. Well, yesterday I learned an interesting quotation by an American comedian named Merz. And the saying was, or his quotation was, "You can't help getting older, but you have not to get old." So I can add now, you must not look old. And that opens the field of cosmetic dermatology. And I would like to talk about botulinum toxin products. Well, this shows you an interesting clinical study. What's the difference between before and after treatment? You can see here before the treatment, especially at maximum frown, does not exist. You see the glabellar fold, and after 4 weeks treatment, there is no glabellar fold anymore. And the difference is, of course, or the treatment is, of course, the injection of a botulinum toxin product, in this case, Xeomin. Well, what about botulinum toxin? What is the substance botulinum toxin? Well, it is the most poisonous poison in the world. There is no other material, no other substance in the world which has a higher toxicity. To give you a number, the human lethal dose is less than 1 billionth of a gram per kilogram. You see a lot of 0, 0, zeros. So it's really a very low amount of protein which is already toxic. That means when you want to develop a pharmaceutical with it, the active dose has to be even much reduced compared to this lethal dose in humans. Okay, what about the active substance? The active substance is a 150 kilodalton protein, which is produced by a bacterium called Botulinum, Clostridium botulinum, an anaerobically growing microorganism which can contaminate food. So these bacteria produce a food toxin. The mode of action, you might know, this botulinum toxin, this protein binds to motor neurons or cholinergic neurons which activate glands, for example, the sweat gland or the saliva gland. After binding, the substance is taken up by the neuron and transferred into the cytosol of the neuron. The next step is the cleavage of a special protein called SNAP25, which is required for the secretory process. And that has a special consequence, the inactivation or the cleavage of this protein. Namely, the secretion of acetylcholine is inhibited so that the muscle is paralyzed and cannot be activated anymore. So that is the action mechanism of botulinum toxin, which is used for therapy of different diseases. Well, on the market there are 3 different products: Botox, Dysport, and Xeomin. These products are approved for several indications, like cervical dystonia, blepharospasm, spasticity, or the treatment of glabellar frown lines in aesthetic medicine. All these products contain the 150 kD neurotoxin, which is the active substance in all the products. Botox and Dysport are special because they also contain additional proteins, the so-called complexing proteins, which are also produced by the Clostridia. In contrast, Xeomin only contains the 150 kD neurotoxin, the active molecule. So what are the differences between the products? Let's have a look to the manufacturing process of the active protein, the neurotoxin. The production procedures are different. For Botox, a special series of precipitation steps is used to end up with the so-called 900 kD complex. Dysport is manufactured by chromatographic procedures, ending in a mixture of complexes. The size is not published by the manufacturer. What about Xeomin? Xeomin is also produced with a series of chromatographic steps, which end up with a pure neurotoxin without any other Clostridial proteins. Well, there is evidence in biotechnology which says that the product is the process or the process is the product. So that means the structure, the activity and other properties of biological products are determined or controlled or influenced by the process itself. Well, this shows you the structure of the botulinum toxin complex, which is in Botox. You see different proteins. These are all the complexing proteins, and this part only represents the neurotoxin. So what we did was that we removed all the complexing proteins and ended up with a pure neurotoxin. And this shows you the crystallographic structure of the neurotoxin with different domains. For example, this is a binding domain, and this is translocation domain, and in this part is the enzymatic activity. So Xeomin is based on the pure neurotoxin with no other Clostridial proteins. Is there any difference in the structure of the neurotoxin of the different molecules? Well, the amino acid sequence of all the neurotoxin molecules in the different products is identical. But we do not know whether the spatial structure, the conformation of the protein, is really identical. And remember what I said, the process is the product. So the process can influence the conformation. We don't know that in detail. This shows you some properties of the botulinum toxin products, Botox, Xeomin, and Dysport. There are other names in the literature given by the FDA. Botox, produced by Allergan, is produced in a process called vacuum drying, whereas Xeomin and Dysport, produced by Merz or Ipsen, are manufactured by lyophilisation. That are different processes. All the processes end up with a dry product, which is much more stable than the neurotoxin in solution. Molecular composition, I already talked about that. 900 kD complex in Botox, 150 kD neurotoxin in Xeomin. What about the amount of Clostridial or bacterial protein which is injected into patients? That is also different. We will come to that later. There's also a difference in the amount of the neurotoxin protein which is injected. And what about the excipients? In all the formulations, we have human serum albumin, and there's a special reason for that. Because the amount of neurotoxin is so low, human serum albumin is used for stabilization of the neurotoxin molecules. Let's talk about the amount of Clostridial protein injected into patients in more detail. When patients are treated for glabellar lines with 20 units Botox or Xeomin or 50 units Dysport, this is the amount of protein which is injected, about 0.1 nanogram of bacterial protein in Xeomin or with Xeomin. The amount of Clostridial protein injected into patients with Botox is about 10-fold higher, and Dysport is somewhere in between. Interestingly, the amount of neurotoxin protein, the active molecule, is also different. Why that? Well, with a specific ELISA, we have analyzed the amount of the neurotoxin protein in a 100-unit vial of Xeomin. It's about 0.5 nanogram. The amount of neurotoxin protein in Botox is higher, about 50%. What is the reason? Why that? Well, part of the amount of neurotoxin protein in the vial is inactivated. It's inactive neurotoxin, which cannot be taken up anymore by the neurons, but what can be taken up by, for example, immune cells. What about the specific activity, or the activity in the different formulations, different brands. This shows you an analysis of 5 different batches of Botox with the standard assay, the so-called LD50 assay, which analyzes the biological activity in the final drug. And as you can see, about 100 units per vial. What about Xeomin? The same, about 100 units of LD50 activity in vials of Xeomin. What about efficacy in the clinical atmosphere? This is the endpoint of a clinical study, a large clinical study with 370 patients in a head-to-head trial with Xeomin and Botox. And as you can easily see, there was really no difference in the final outcome. The response rate was about 96% for Botox and for Xeomin. The same was true after week 12. And this is a result by the independent rater. The investigators and the patients came to the same conclusion. No difference between the products. So there's a 1-to-1 ratio between Botox and Xeomin. What about the duration of effect? Is it different? This is analyzed in a study with 180 patients treated with Xeomin, Botox and Dysport. And the result was the following. As you can see, the duration of effect with Xeomin, 146 days, very similar with Botox, and the same, in the same range, Dysport. This is for female patients. Interestingly, for male patients, the duration of effect was shorter, but there was no difference in the duration of effect when you compare the different final drugs. So duration of effect, no difference. Let's come back to the complexing proteins. They have no role in the activity, but they can have, and there are claims, could have an effect in the therapy. That means protection against degradation, stabilization, or inhibiting of the neurotoxin migration into adjacent tissues. Why is diffusion interesting? Well, adverse reactions of botulinum toxin therapy depend on the movement, the migration of neurotoxin molecules from the injected muscles into other muscles. The movement of proteins is dependent on the size of the molecule. That would mean if there would be a 900 kD complex, the movement would be slower, or the rate of adverse events would be lower. Is that really the truth? Well, it is not, and that is demonstrated in this analysis. These investigators have analyzed the different protein species in vials of Botox, Xeomin, and Dysport, and this was the result. They found that mostly free neurotoxin was present in the Botox vial, and only a little bit of a smaller complex. The rest were complexing proteins without neurotoxin. The same was true for Dysport. In Xeomin, of course, only the neurotoxin. This was confirmed in a clinical study in which Botox and Xeomin were injected into the forehead of patients, 5 units of each product, and then with a special stain, the area of anhydrosis was made visible and measured. And the result was no difference in the area of anhydrosis. That means it's really true. There is no difference in the diffusion of the different products. What about stability? Are complexing proteins necessary for the stability? Definitely not. As you can see here, Botox and Dysport have to be stored at 2 to 8 degrees, whereas Xeomin can be stored for 4 years at room temperature. And this is very interesting for biochemists. Storage at 60 degrees for 1 month did not reduce the biological activity. So it's a very stable product. So there's no reason to inject complexing proteins into patients, but there's a scientific reason not to inject scientific proteins. And we come to the formation of antibodies against botulinum neurotoxin, where the formation of antibodies terminates the therapy with botulinum toxin, because then secondary non-response is induced. So what is the critical factor for the production of antibodies against the neurotoxin? Where the immune system must be activated. The dendritic cells need a signal to activate T lymphocytes and then B lymphocytes. As you can see here, there are different signals which can really activate dendritic cells to process the antigen and to secrete cytokines. That is, for example, DNA, that is flagellin, that are components of the cell wall, and other substances like lactans. So, which factors can play a role in the stimulation of immune response against the neurotoxin? Well, there are hemagglutinin, there are other clostridial proteins, and there is inactive proteins, neurotoxin, which I have already mentioned. What about hemagglutinin? Well, hemagglutinin are lactans which can bind to immune cells, and that they really can bind is shown in this slide. Immune cells were incubated with complexing proteins, and all the different spots you see on the right-hand side represent binding events between the complexing proteins and the immune cells, whereas the neurotoxin itself, botulinum, in the left picture, did not bind at all and cannot activate the immune cells. So in consequence, there is a difference in the immunogenicity when you compare complexing proteins containing products with products which have no complexing proteins. That means Xeomin. Okay, my conclusion: products with complexing proteins and Xeomin show the same efficacy and longevity, duration of effect in clinical practice. There is no difference in the migration characteristics. They have the same adverse events, but Xeomin is much more stable than the other products, can be stored at room temperature. The amount of antigen injected into patients is the lowest with Xeomin, and Xeomin has a low risk of stimulating antibody formation. Thank you for your attention.
Thank you, Dr. Frevert, for the excellent talk. Are there any questions for Dr. Frevert? Yes, sir.
Yes. Yep. Yeah, we are doing that. That is the next generation of botulinum toxins, where we modify the amino acid sequence, but the products on the market are based on the wild-type Clostridia, which were isolated from the environment. And all the products are based on these proteins. But the purification procedure is different, and for some reason, Botox and Dysport still have the complexing proteins, because the companies thought these proteins are necessary, as I have mentioned, for stability, for inhibiting diffusion, spread of the molecules, and other reasons. But that is not really true, as I have shown you. The pure neurotoxin, without any other proteins, is active, it's stable, it has the same diffusion characteristics. There is no difference. But the difference is that the complexing proteins, which form this complex, have an effect on the immune system.
So it would be much more easier to express in other system, and even cheaper than to produce.
Would be cheaper, but you must also purify. When you produce it in E. coli or in yeast or other organisms, you must also purify.
You can add a tag, for example.
Yeah, well, a tag is not enough. I don't know if you have experience with tag proteins.
A little bit.
Usually the purity is about 50% after using this affinity chromatography with the tag. And there are a lot of tags like his tag and so on. You can use that, but you also purify or separate a lot of host cell proteins, which you have to remove after that with different chromatographies.
I've experienced in plant cells, so the expression of the proteins. And there are several ways to purify, really
Yeah.
purify 99% pure, 99% pure protein from other organisms.
Yeah,
So that's
that would
why
be-
I'm asking,
yeah.
yeah.
That would be a new approach. As far as I know, botulinum toxin was never expressed and produced in plant cells.
If you would
Could
like
be
to try.
interesting because in plant cells the protein might be glycosylated, possibly, which could add a couple of interesting new properties.
Thanks.
Continuation with the same question. Have you considered using the CHO cells?
In
CHO
what?
cells, Chinese hamster cells, ovary cells.
CHO cells.
Uh-huh.
No, that was never tried. It was well, we produce it in E. coli,
Uh-huh.
and there
Yeah,
are
but I mean-
also publications concerning expression in yeast cells,
Yeah,
but
actually,
never in CHO cells. I don't know what would be the advantage to have it in CHO cells.
CHO cell is already FDA approved, and if you have a concern with the glycosylation when compared to the bacteria E. coli, the CHO cells is a mammalian cell line, so you will not be having any problem with the glycosylation, any post-translational modifications.
Right. Yeah.
So that's one advantage, because for some of our work to make the recombinant proteins, we prefer to use the CHO cells rather than any other system. So
But-
its purification, just one step, affinity purification is more than enough to get up to 90-95% of purity.
Yeah, we can also have this purity, of course. And from my understanding, E. coli can be cultivated much easier than CHO cells. You can have a very high density of E. coli and have high yields of the product. So I would prefer E. coli unless, and that's the point, unless you want to glycosylate.
Glycosylation.
That would be an advantage, but you can also glycosylate in yeast cells, for example, or you can do it in plant cells or in fungi. That would be an
Mhm.
alternative, but CHO cells is an alternative, yeah.
Okay. So my another follow-up question is, with your fluorescent microscope studies, you use this lymphoblast cell line.
Pardon?
In your
okay, yeah.
lymphoblast cell line, right? So that is lymphoid in origin. Have you checked with the myeloid origin, like dendritic cells or macrophages? Because those are the
Yeah,
one which
yeah,
is
sure,
antigen-presenting cells.
That is an
So-
ongoing project right
I
now
see.
to analyze it really with human dendritic cells, which can be isolated. That would be the next step. So this was the first experiment to really show that there is an interaction. But it has to be shown that this interaction is also specific in human dendritic cells.
I see. Okay. Thank you. Excellent.
Thank you. I have one question for you, Dr. Frevert. You have showed that Xeomin and Botox are equipotent. The way you dilute it, is it similar?
Yes.
Exactly using
Absolutely.
normal
Similar.
saline
Yeah.
and...
It's reconstituted in normal saline, all the products. And, well, you can choose what volume you use. But in these head-to-head studies, the volume was identical, because the volume can also have an impact on the diffusion, on the spread of the molecules. So in all these head-to-head studies, which show that there's a 1-to-1 ratio, the dilution was identical.
Thank you very much. Thank you once again, Dr. Frevert, for the excellent talk.